Professor Karim Fouad. Professor Fouad is a professor and Canada Research Chair at the University of Alberta. He studied biology at the University in Constance, where he also completed his PhD in neuroscience in locomotor control. He continued with postdoctoral training at the University of Alberta in locomotor control and then at the University of Zurich, where he began to study neuroregeneration and the role of neuroplasticity in rehabilitation training in animal models of spinal cord injury. He started his laboratory at the University of Alberta in 2001, where he focuses on strategies to promote repair and recovery after injuries. Dr. Fouad received support from a wide range of founders, including CIHR, Wings for Life, ISRT, the Craig Nielsen Foundation, and the Christopher and Donna Reeve Foundation. Please join me in a virtual round of applause to welcome Professor Fouad.
Well, thank you. Well, and thanks everyone for attending and for having me. It's nice to, well, at least see Miloš's name. We have a long history, and so it's my pleasure to see you all here. I'm going to share my screen. And I hope you can see this.
Yes, we can see it.
Good.
It's not in slide mode.
I'm not there yet. There we go. Now.
Perfect.
That should be all good. All right. So I'm trying to really address this talk and focus a little bit on rehab, but nevertheless, it's still going to be somewhat of an odyssey of my life, and it kind of tells you a little bit about the evolution of my research and how I came to where I am now when I study, among others, rehab training in animal models of spinal cord injury. So, because I always forget at the end, I just want to acknowledge this. Since this has been an odyssey, there have been many collaborators. And on the left, you see all the students, on the right, the collaborators and my main funders. And I start here, and that's actually work from the time when I met Miloš the first time in Zurich. And the point was that we tried to promote regeneration of axons in the spinal cord. And as you probably all know, this is not very successful. Here on the left, let me just go to a laser pointer. You see actually neurons, axons of the corticospinal tract in the spinal cord, and here's the lesion site. So that's the lesion site. And you see that they all kind of form these little ink blots at the end. These are retraction bulbs. That's basically when the neuron grows and it meets one of these detour signs or stop signs on the scar or in myelin, then they withdraw and they form these retraction bulbs. So that's the situation. There is no real regeneration, regrowth in the spinal cord. And this is due to many different inhibitory signs or challenges neurons have to overcome. So it's not just one. There are many, many reasons. And we were kind of struggling so much, or have over many years now, to promote repair because there are so many signs that tell neurons not to do so. But one of the questions we had is, what is the remaining circuitry doing after an injury? And we know that there is moderate recovery, and you can even amplify it with training. So how is this happening? What is going on? And I really wondered that if we can understand this naturally occurring moderate recovery, maybe we can use that knowledge and then actually promote more substantial recovery, rather than overcoming all these inhibitory signs for regenerating neurons. So we basically looked in a model where we labeled corticospinal tract neurons in the brain of a rat. We cut the corticospinal tract unilaterally, and then we looked, what's happening now with these neurons? And one thing that we found, And this is basically here axons that are cut down a horizontal way. So a horizontal section of the corticospinal tract, white matter to the left. This is gray matter. And they were traced in the hind limb area of the motor cortex. And they run basically down to the lumbar enlargement. But we look here in the cervical enlargement. And there are not many, but there are a few branches that exit. So this makes a lot of sense because some neurons in the cortex probably innervate the arms and the legs at the same time. So there are a few collaterals here, but not too many, as most of them probably branch in the lumbar enlargement. If you do an injury, let's say down here somewhere, you do cut them, you see that there's an increase in these collaterals. There are more branches growing out, although they were supposed to go and innervate motor neurons or interneurons going to the hind limbs. Now in the cervical enlargement, they sprout. And you see here a little bit enlarged, they form these clusters and seem to target neurons, especially in this area. So we're wondering, what are they doing? Are they going to motor neurons now, or are they maybe even going to interneurons? So one of the ideas was that in the spinal cord, there are always interneurons that project from the cervical to the lumbar enlargement. So an idea was that maybe we have the following scenario. Here's our corticospinal tract neuron, and we cut it, so it gets injured here. Obviously, what happens now is that this part dies off. That's the Wallerian degeneration. And then I showed you these branches, and maybe these branches go to interneurons that are spared. Maybe interneurons that project all the way down to the lumbar spinal cord. And that was kind of a little bit wishful thinking at the time, but we basically tried to prove this by giving a red dye in the lumbar spinal cord. So everything that still projects to the spinal cord, lumbar, would turn red. So this guy would turn red if it projects all the way down. And then we traced these in black and said, okay, let's look whether there are these connections. And that actually happened. So here's a red interneuron that is projecting all the way down from the cervical to the lumbar enlargement. And here you see a corticospinal tract that kind of was wandering around until it found this neuron, and then it connected to it. So that was kind of stunning. Although we don't have regeneration, there's this possibility of rewiring the spinal cord. And with that knowledge, I really thought, well, how does this plasticity and training go hand in hand? I always wonder when I think about training, what's the chicken and what is the egg, right? Is such kind of neuroplasticity the key to enable training? Or does training maybe increase this plasticity? And at the end, I can tell you already now, it comes out that's probably both is true. Oh, so rehabilitative training helps recovery. We know that. You know that very well. And that rehabilitative training is really based on plasticity is a given too, because plasticity is any adaptation that we see in the spinal cord. And so we need adaptations to actually respond to training. So can we understand this a little bit more? That was my big goal in my research. And the first experiment that I did was we took animals, rats, with a thoracic spinal cord injury and put them on a treadmill. And we want to see whether this training improves their recovery. For that, we used a score. It's called the BBB score. And that's many years ago, right? And you see that the animals over days, they recovered and the training didn't make them better at all. Now that was devastating because we trained about 30 rats, 20 minutes per rat a day. That was a lot of work. Actually, the one who was untrained, so it was only 15 that got training, but every day, and no effect. So I was wondering hard about this. Why is there no training effect? Because we knew already that training in humans and in cats improves locomotion. So one of the ideas was that, well, rats their belly is basically on the ground already. The center of gravity is so low that when they walk, even when they're paralyzed, they're dragging their legs and provide them with sensory stimuli. Also, they provide them with a sensory stimuli like you'd be on a treadmill. So the idea was then, well, maybe 20 minutes of us training animals stands in no relation to the whole day that they move around in their cage and basically train themselves. So when I discussed this with a colleague, David Magnuson, we agreed that, yeah, probably the spontaneous activity is what trains them. And he did an experiment that I wasn't willing to do that. So he did it for the greater good here. He actually did put rats in a little wheelchair. Basically, after injury, he strapped the hind legs in this little cart, and they were not able to move the legs or to train them in stepping. And I want you to focus only on the green and the red group. So you see that the animals got a lesion, 0.0. The first test, they barely could walk. They're probably still in spinal shock. And one week after, they bounced to a score of about 11, and then they continue to recover to about 18, and 21 is the max of the score. So they're pretty good. They're recovered close to normal locomotor ability. Animals that were strapped in here in red, well, they got worse and worse and worse and worse. And then they stayed at this level about 4 or 5. And when they were released from the wheelchair, they recovered a bit, but they never recovered at the same level as those that were moving around. So that was a big lesson. So spontaneous activity, this training, is very important in the animals to recover. And just sitting in a wheelchair, or lying in that case in the wheelchair, doesn't allow you that recovery. And when you don't do it to a certain time point, you will never recover to the same level. So there seems to be also a window of opportunity here. So that was a pretty big finding. And for us, it was a sign, okay, training, maybe locomotion is not the right thing to do. So we went to a different task and we engaged them in forelimb reaching for little food pellets. And you can see one here. And in the initial phase, and when the score or this training was developed for rodents, actually with stroke, the pellets were given with a little forceps. And reaching for such pellets is something they wouldn't do in their home cage, which is handy, so we can actually train it without getting distracted by self-training. And if you cut the corticospinal tract, it's very important for that fine, skilled movement. So it's a good thing to really, if you want to study corticospinal tract plasticity, this is a good task to look at and see whether it's involved actually in recovery. A little bit after starting to train with these pellets, with giving them one after the other, we were getting quickly tired of that. And we developed a little bit of a robotic device where we had a pellet dispenser built, and it could push, you see the pink thing is an arm that pushes the pellets up through a hopper. And so we could push a button and then dispense these pellets a little quicker. That made life for us easier, so we can actually start to study the effect of rehab training in rats. But even that is still a lot of work. And the problem is also you train the animals when you want to train them, not when they want to train. So we thought a little further how we could automate this whole thing. How could we automate training? And we built this little automated chamber where animals would come in, and there's a floating floor with a sensor underneath. And as you see, there's actually an RFID antenna. So they have a microchip, and we can know which animal is entering, how heavy is it. And once an animal entered and we identified it, the little robotic device would basically deliver, you see on this arm, it goes up and it delivers a pellet to the animal. And then we have various sensors that could actually measure if the animal was reaching forward, if it actually removed the pellet, and if the pellet dropped either down or whether they could retrieve it. So it was all automated, and we could basically let the animals train themselves. So this is how this looked at the end. We had all these cages, of 4 or 5 cages connected, and they were attached left and right. They had robots to train. I think on the other side we had 2. And the animals could self-motivated train themselves around the clock, right? They're a little bit more active during the night than we are, so they could do whenever they wanted. Obviously, one problem was that it had a little bit of variability, because it comes out not all the animals trained the same. And so the first thing you're going to say, oh, we could have told you that, is that we saw that the training amount, how many pellets they take, matters on how good they are. So the final SPRGR, it's basically the success rate in the reaching and retrieving the sugar pellet and eating it. And that's the total number of attempts. So animals with lower numbers of practice, they didn't do so well, whereas a group that trained on a higher number of attempts, they did much better. And we used the cluster analysis to actually detect 2 groups, because we wanted to look at patterns of how they train and whether it mattered. But for now, while we know already more attempts, better recovery. And importantly, that's not a linear relation, right? There's an area where you get most bang for the buck, but at a certain point, they don't really get much better anymore, right? So it seems that, above 10,000, they're not really gaining a lot of success anymore. So that's important when you want to design effective training. So then the other thing we learned is that the intensity matters. First of all, on the left, we kind of looked at, how many hours did they train with how many attempts? And we saw that they actually work in these bouts. They train, and only in a few hours they work very intensively and train very hard, and others they don't barely go in at all. Which kind of makes sense. Once they're in the robot, they kept going for a while and worked hard. When you plot the recovery again to the number of attempts per hour, so the intensity we define as number of attempts per hour, you see also that there's a very steep increase to a certain intensity, let's say 50 per hour, and then we basically plateau. But if you train at lowers, you don't reach this ability. So not only does the amount matter, but the intensity. And when we looked at the animal groups that we identified before, remember these 2 groups here, the low trainers and the high trainers. If we look at this again, we could see that all the high trainers actually trained at a much higher intensity. So this is logarithmic scale. That's probably somewhere around 75 per hour. That these animals that did so much better and recovered so better trained at a much higher intensity than the others. So importantly, although I talked about a window of opportunity for training in the other study that I showed you from Magnussen's group, we had by chance, and we cannot explain it, 2 animals here that didn't really train a lot. You see the accumulated attempts here over time. This is days. So this is over a month, 50 days after. They haven't really trained a lot, not a lot of attempt rates. Their success was also pretty lousy. They weren't very successful in doing it. But then suddenly, and it looks like the success went up first, suddenly they started going like crazy. Maybe they were motivated by a little bit more success, and then they started increase their attempt rate. And so even 50 days after, in that case, the increased intensity improved their recovery dramatically. So it kind of shows that, you don't really get a lot out of this low intensity training. It is the intensity that really adds the spice to the training. So then obviously we're working with rats. We have the advantage. We can now try to link this training to any kind of anatomical plasticity. And we stuck to what we did before. We just kind of modernized it a little bit. We have again a unilateral lesion of the corticospinal tract. We can trace the cortices both sides. One we do with a green dye, with a virus vector actually, and for green fluorescent protein. And the one here is reddish, a TD tomato is the vector expressing. And this is the dorsal column here, right? That's the cross section of the spinal cord now. Here's the dorsal column, dorsal horns, ventral horns. And now you can look at this little window here. You see where the corticospinal tract sends its collaterals out here. That's pretty much the same that we have counted before. Collaterals coming out from the tract into the gray matter. And you look, when we look at low intensity and high intensity, it becomes pretty obvious very quickly that there is a high density or a higher density after training. So we could actually show and quantify this then, that training increases collateral sprouting beyond spontaneous sprouting after an injury. So that's good news. We have an anatomical link that basically links training to plasticity of the corticospinal tract. So now a big question is, does this matter? Does this relate at all to recovery, right? I mean, there it goes up, these collaterals, but does it matter? And we did kind of a complicated experiment for this, so bear with me. Once again, we trained animals in the palate reaching. We did a lesion of the spinal cord, again, unilateral corticospinal tract lesion and little rubrospinal here. So we cut the whole corner away, and then we trained them. Then we see how much did they recover from the training and how, you know, compared to untrained animals. And then we went up a little rostral into the brainstem and cut specifically the pyramid. So we cut the corticospinal tract at the level of the pyramid, cut that off so that the entire corticospinal tract is now gone. So basically, we cut the corticospinal tract here and here. So after the second lesion, we remove all this part. So any sprouting that I showed you before that is above the injury, sprouting to interneurons or motor neurons, whatever it is, any of that sprouting now is removed with the second lesion. And we can see how effective this was and if there's a link between this corticospinal tract plasticity that happened in here or not. So that's the result of that. So first, here's the success rate in reaching with training and no training. So this is with training. They went nearly to 90% of baseline. Without training, they stayed at 50. So that was a really good experiment in training efficacy. They went 30% better with training. That's quite something. So then we did the second lesion. Now, in untrained animals, we basically collapsed down to 0. Animals were unable to do this. But more importantly, these animals dropped even more, right? I mean, they had gained more recovery, but they lost that gain much more, right? This is, what is this? This is from 50, 10 to 50, roughly, so 40. But that goes from 80 to 20, so it's 60%. So this is dramatically more that we lost because now we removed that plasticity in the corticospinal tract above the injury. Interesting, there's also something left, which makes sense because when we train, we're not just training the corticospinal tract. Plasticity will probably be seen on other tracts like the rubrospinal tract or reticulospinal tract. So this is probably what that is here. There are other tracts that also underwent plasticity and the animals recovered better. But I think from that, I can really conclude that we can link this plasticity that we see, that sprouting, to recovery. So this sets the stage, right? What now? What can we ask now? And this is actually a question I have for you. When you work in the clinic, what are the questions you want to ask now? I mean, oh, I'm out of paper. That's not good. Here we go. we have now a model to study rehab training and plasticity, how they link. We can ask, how does training promote plasticity? What are the molecular mechanisms? And can we enhance plasticity then when we understand these mechanisms, these naturally occurring mechanisms? And then we could ask question, so does training actually, the efficacy decline after injury, and why? Can we actually restore training efficacy in chronic injury? That would be important. So we have now the whole playground, and we can go in, and we started that. And one was, for example, to say, well, we know that with training, growth factors like BDNF, brain-derived neurotrophic growth factor, they get upregulated. Is that the reason for increased plasticity and training efficacy? Is that the mechanism? So we said, well, let's just give BDNF to the cortex and see what that does. So we basically had a whole bunch of animals, and then we counted to collaterals again, and we had animals without injury, a vehicle treatment, BDNF alone, no lesion. BDNF didn't do anything. There was no increased sprouting. And even the lesion didn't really increase a lot of sprouting in this experiment. But if I give the lesion and BDNF, there was a substantial effect. We nearly doubled the number of collateral sprouts. So that was, at the time, confusing. Why does BDNF not do anything without the injury? And we will come back to that. The next experiment, we said, oh, we have to probably see whether this links to recovery and whether training is really important to translate that recovery. And we added another group. We did have NT3 that we gave, it's neurotrophin factor 3. We gave that in the spinal cord. BDNF, again, we gave in the cortex. And then we had animals with training and without training. So this bar here is baseline. That's how the animals performed before in the reaching task. These were animals here. They just got GFP. It's basically a control treatment, no training. We added training to the control treatment, and there was a little bit of a training effect. We gave BDNF NT3 alone without training. There was no recovery. If we added training, they recovered better. But if we only gave BDNF, the recovery with training was even stronger. So that is an important lesson once again, and others have repeated this lesson. So if we give treatments to increase neuroplasticity, we actually need to add a training paradigm to translate that plasticity or sprouting or whatever it is into something meaningful and into recovery. So that was a very important lesson that we learned at that day. But then I kind of was running a little bit out of steam, and we were thinking and playing with some data, and we kind of pooled data. And that's, for example, data from 3 experiments, all animals, some trained, some untrained. And curiously, 100% here is baseline. So that's animals, how they performed before, and that's how they performed then after injury and or training. Now, if we cover this up, everything under baseline, you see that animal, there's actually a bell curve here with animals with a small lesion, and especially training, they seem to be better than before. Now that's curious. How could that ever be? Could potentially the inflammation caused by a small injury promote neuroplasticity, and therefore training has a more fruitful ground to really rewire the nervous system in a meaningful way to improve the reaching task. And that brings us back to the experiment with BDNF. Remember when I said that BDNF didn't work alone, it needed the lesion. Maybe with a lesion, there's inflammation that plays a role in enabling neuroplasticity. And that could also explain why after injury, training efficacy is reduced, because maybe inflammation has declined over time and therefore training is not as efficient anymore. So these were all little pieces of information that we thought that maybe inflammation plays a major role in training efficacy. So then when Abel Torres' was in my lab, we designed the following experiment. We took animals, we trained them in the reaching task, and then we did an injury. We waited for 8 weeks, which in the rat is considered pretty chronic. Then we actually triggered inflammation. We injected IP, LPS, so that's lipopolysaccharide, basically bacterial cell wall, and we gave it at a very low dose. The animals basically had 1 degree of fever for 1 day. So they were basically pretty happy the next day, and they could start training. And that's when we started training daily for 7 to 8 weeks. I think we did 8 actually. And then at the end, we did tracing and analysis and all that. So first of all, people are always concerned that while they were sick with LPS, they didn't train. But keep in mind, LPS sickness lasted only for a day. So when we looked weeks of training here, the attempt rate of how much they trained was very, very similar between the orange and the blue. That's the cell line training and the LPS training. So these are the two groups we can compare to each other. This was LPS alone, cell line alone with no training. So we didn't have data before. Now, when you look at the success rate in reaching, you see that pretty soon, so 3 weeks after the LPS injection, the training became more efficient. We had better success rates in these animals very quickly. That was quite stunning and repeatable. We could show that again, but one thing was more striking. When we train, and you know this probably very well, many times what we train is that a compensation is trained to actually deal with a task. In rats, it's very typical when they lose their digit function, they start scooping, and it could well be that the training really improved success rate because they learned how to scoop more effectively, but not really use their digits and hold on to the pellet. We could test that. We introduced a little gap here. So we cut a piece out. So if an animal would drag the pellet towards itself, the pellet would fall down and it would not count as a success. When we did that, then we saw the following here on the right. So on the left is basically the summarized data from what I just showed you before, where animals with training got better, but with the LPS, they got even better. But now when you look here, trained animals are as bad as untrained animals, because at the end, we just trained them to scoop. We trained them to drag the pellets toward them, and they got quite good in it. But the animals with LPS actually showed a sign of true recovery, and that they could hold on to these pellets and retrieve them more successfully than all the others. We could link this then also again to our plasticity. And here we analyzed it a little bit more sophisticated. We used heat maps, and the more red it is, the more sprouting, the more collaterals, the higher density corticospinal tract fibers have in the gray matter. And when you see the vehicle group, that's the projection that we had. With LPS, the projection seemed to change a little bit. There seemed to be more sprouting overall, but also especially in this area that really didn't have anything in vehicle treated groups. Vehicle and training look pretty similar, a little bit increased sprouting, but the same areas. And when you give LPS, that projection seems big time increased, especially here in the intermediate areas of the gray matter. So basically what this says, it could indicate that LPS actually does increase sprouting, but when you add training to it, you basically direct the sprouting towards a meaningful target and rewire it in a different way. So really, what training seems to do, it is harnessing the plasticity that we could unleash with inflammation. So at that point, I thought, oh my, that would be great. If that's true, then maybe I can do this in healthy animals, because then I could, everyone who would like to learn tennis or golfing, we could sell an inflammation booster and you could do much better motor learning. And this idea, as great as it was, it failed as miserable. And it led me to a totally different stream of research recently. And that was that when we had this, my students come in and says, well, I know exactly what animal got the drug because they seem all anxious. And we tested that then, and it comes out that if you give LPS IP in healthy rats, and you test them on something we call the elevated plasmase. This is basically an arena where the animals can walk around freely, and you put in one animal at a time, and then it can walk either on a runway that's in the dark here, nice and covered, or in the open. And an animal, a rodent, they don't normally be elevated, and they don't normally want to be a lot out there, because this is dangerous, right? Anytime a bird could come down and hunt them. So there's a certain ratio that they actually would go and explore, but most of the time they would stay inside. And you basically look at the time, how much time they spend in the open, and the less they spend in the open, the more anxious these animals are. And you see that LPS decreased the time in the open quite a bit. So inflammation seems to be increasing anxiety. And now we were a little curious. We had this glorious study before where we showed that LPS improved training and reaching, and now we say it improves, it increases anxiety. Why did we not see that? And we had a couple of theories. Maybe spinal injury prevents the effects of LPS on anxiety. Or maybe they were so subtle we just didn't see it. Or maybe rats with spinal injury already exhibit anxiety-like behavior. And it comes out that anxiety and depression are always associated with injuries and damage of the central nervous system, and therefore also with inflammation. And so there are quite a few studies out there showing that there could be a very strong link. So we were thinking, okay, let's test our animals with spinal injury. Does spinal injury in rats increase anxiety? We used the same apparatus, and first we looked, is the distance of travel changed? Because you could argue immediately, say, well, these poor animals have an injury, they can't walk. But they walk quite fine because it's a very distinct corticospinal tract lesion, it doesn't really impede their walking. And you see that the walking distance between these two groups was the same in the field. The issue was that the sham animals, they only walked, well, 20% exploring, whereas the injured animals, they didn't explore at all. They basically stayed pretty much in the darkness. So they were very anxious. So we're not the only one to show this. Others could now repeat this. Comes out that the spinal injury triggers significant anxiety. And well, we wouldn't be researchers if we wouldn't be interested in how that happens. And we were thinking quite a bit. And by chance, a friend of mine, Phil Popovich, so there's another Popovich just with a CH, he did publish a study in a Journal of Experimental Medicine where he showed that there's a gut biosis that interferes with recovery after spinal injury. And a gut dysbiosis after injury is not necessarily a surprise, right? Because the gut is also innervated and very much affected from a spinal injury. So that is not necessarily a surprise, but the surprise is that a gut dysbiosis can lead to a leakage in the gut-blood barrier. And when this barrier function of our intestine has changed, it can add to leakage of things like LPS, bacterial matter, viruses, into the bloodstream and promote inflammation. And by that, it can affect recovery. It could even exacerbate injury and potentially also trigger the inflammatory induced anxiety that we have seen. So because this was done in mice and with a different lesion, we repeated that in our model. And just looking at the Petri dishes before an injury, day of injury, 3 days after, you see there's a dramatic change. And within 14 days, it slowly comes back. Still a little bit too much E. coli here, but it changed. It seems to recover after 2 weeks. So we see the same. Even with a small cervical spinal cord injury, we have this change. Now, the big question was, could such a change actually affect inflammation and therefore anxiety, for example? So we did, I always call it, we did a shitty experiment. We gave a spinal cord injury after training animals, testing them, and various outcome measures, collected before an injury poop while they were healthy, extracted the bacteria, and after injury, we fed them for 3 days these bacteria. We collected again and also later, and we sent all this data for sequencing to see whether our treatment actually affected the dysbiosis. And so I can tell you it did affect the dysbiosis. I didn't want to bore you with some sequencing data, but the coolest thing was that we actually have influenced anxiety dramatically. And you see this, here's the total distance traveled between all our control animals, sham lesion, the spinal cord injury, and the fecal transplant. And these are healthy animals. So they walk pretty much the same distance after spinal cord injury here, these two groups. But if you look in the times in the open arm, once again, the spinal cord injured animals barely don't go out in the open arms. And you see this up here, that's just an image of how much they would have been in the closed. They walk all in here. Whereas when you treat them, you see them in the open again, similar to what you would see in healthy animals. So really, by just addressing the microbiome, we could actually restore or prevent that change in anxiety-like behavior. And this really drives me to my summary at the end and bringing a little bit the problems with spinal cord injury, whether it's in animal models or in humans, right? It's so hard to predict outcome. And we struggle with this because we constantly underestimate all the factors that contribute to recovery, right? And one is, we know the corticospinal tract can rewire and it contributes to recovery, it can. But we don't know much about other tracts. We've studied a little bit reticulospinal, propriospinals. We know all these other tracts also undergo plasticity because we just focused on the corticospinal tract because it is so easy to do. This rewiring can be boosted by training. We know that too. We showed this. And inflammatory components clearly contribute to plasticity. And that's not necessarily a good thing because the plasticity associated with anxiety is not a good plasticity. And it's linked to inflammation. And then things as simple as the microbiome changes, they contribute to inflammation. And when we look in the figure here, this is where we see all these factors that basically link us to how do we recover, right? What's the lesion size? Then interspinal modifiers, like was there bleeding? What's going on at the lesion? There could be secondary damage ongoing. Then other inflammatory factors like the dysbiosis or acquired infections. And they all affected this constant and progressive recovery. And I think it's because of all these different factors that we have such a hard time predicting outcome. And this doesn't make it easier for us when we try to design the ideal studies, because we try to restrict our studies so much, and then they would be translated to the clinical setting. And as you well know, there's so many more factors that are not controlled as in our models. So I think this is where we're standing right now. I believe that from our data, there's a lot to learn about training. And I think we have a model that, you know, we could address questions that you might have and that you cannot address in humans out of various reasons, obviously, and also ethical reasons. You cannot just not train someone, but we can ask questions and test things that can be barely done in the clinic and gain more insight. And considering that training is really the one treatment that has translated so well in many aspects from animal models to humans, I think we're having a good model in our hand. And I would be really excited when clinicians would come and start asking these questions to us and say, hey, how can we work together to address these questions? And that's all I had to say. I wanted to thank you for your attention, and I hope there are a few questions for me.
Wonderful, fascinating talk. Thank you. That was wonderful. People in the audience, if you have questions, you can type your questions in the Q&A box, and Dr. Fouad will answer, or you can raise your hands and we can unmute you so you can ask your questions.
That would be preferred, by the way.
Yes, absolutely. So while we're waiting for people to, oh, actually, there's already one. That's Milos. Okay, so Milos, let me just unmute you. Milos, can you unmute yourself, or Michael, can you help with that? Does that work? Milos, is it possible for you to unmute yourself? Does it allow you? Does the system allow you? Or Michael, can you help with that? I don't know how that works. So I guess I'm waiting for Michael to try to unmute Milos. So, or maybe Milos, I'm not sure how to do that. If that doesn't work, can you please type in your question in the Q&A box? Okay, so the question is from Dr. Milos Popovic, and it is, what is the future of biome experiments? Where do you plan to take them?
So the future had already begun. The first thing, people don't like the fecal grafts. So one thing we wanted to test, what does probiotic do in our hands? And that was actually quite a disappointment, because it comes out that probiotics are not very quality controlled. So we had to realize that not because they write something on it, you have the same thing in there, because when we sequenced it, it didn't really contain the bacteria we wanted to see. So we had an entire experiment that kind of went bad with the probiotics. I also think probiotics will not be the greatest thing, because probiotics take a while to act. Because what they do, they're not replace, or they're not bring bacteria that you actually want in your biome. They're basically suppressing the dysbiosis so that the good bacteria that you want, they have a better chance to be restored. But what we're going to do is we're going to continue with the probiotics and want to test this again with a more controlled probiotics. But we're also going to do another experiment where we want to actually see, can we increase the effectiveness of rehab and even the engagement of animals when we prevent the anxiety? Would they engage better in training? And is the effect overall, do we see then a better motor recovery with that? And yeah.
This is great. Can I ask a silly question? Do you think that the exercise will also change the biome? Is there
not a silly question. I'm quite certain it probably would at a certain degree. That's a good question to see whether, especially depending on the intensity, right? When you go really intense, because one thing is clear, stress changes the microbiome. We know that. And that's the thing. Even in our control animals, microbiome changes were seen. The question is only, how bad is the change? And that's something we have to figure out, right? At what point, how big has the microbiome changed to be that the blood-gut barrier actually breaks down and you see products in the blood from the gut? And that's something we need to figure out too. But I'm very excited about these experiments because this biome experiment has been one of the strongest results I've seen in my entire career. Like, it has been so clear and so significant that I'm stunned by it. And I'm not going to give up on that just because by probiotics being a little bit badly controlled. But it's a good lesson too. So we're going to play with this. And I like your question. The intensity could probably change the microbiome.
Thank you, Karim. This was a very nice lecture. I really appreciate you talking about this. This is different and very exciting. Congratulations.
Thank you.
Others, if you have questions, please raise your hands or type in the Q&A box. I'm going to follow up on that. You touched on this, but have you done experiments or are you doing experiments to look at the effect of changing biome or fecal transplants on the rehabilitation progress?
No, well, we did start one experiment and we learned another lesson, but it basically failed. We have to repeat it. The lesson we learned is we gave the biome from animals that were actually highly anxious. They were shipped and had trouble in the shipments. They were stuck in the airport. And we were naive. We just took their poop and said, these are uninjured animals. And it comes out when we grafted it, we made actually our animals more anxious with a poop from anxious animals. So it's a little bit iffy. And you look at many of the studies done in humans, there's not a great quality control on fecal grafts. So I think if you don't have any acute immune system disease, you qualify. It doesn't matter. So it is interesting to see how the screening for what do we really need in a treatment, how that's going to go forward.
So how would you imagine if you have perfect control and perfect unanxious animals, how would you imagine it would go?
Well,
like what would be the hypothesis?
Well, in an ideal world, when we do a couple more experiments, we can probably use AI and figure out what bacterial and components are essential, right? And then you could target those. So lactobacillus is always one of the main players that people want to work with. And we could see that we could have a very simple probiotics. And then we want to use it in animals with injury and undergo the rehab. And I'm quite certain they would be better engaged in training, maybe more focused.
Very cool. Thank you. We are closing on time. It's 1 o'clock. It's if there are more questions, please type them in now. Otherwise, we're going to say thank you to our speaker. Okay, so thank you again for a wonderful presentation. Quite fascinating. I greatly appreciate, and on behalf of everyone in the audience and the Institute, thank you again for accepting our invitation.
You're very welcome.
And everyone in the audience, please join me in a final round of applause to thank our speaker.
Okay. Basically, have a good Christmas, I would say. All the best. Bye-bye.
To you as well. Bye.